Hello, everybody. In this lecture, we're going to be talking about genes and their evolution, or in other words, population genetics. And we're going to also address several polymorphisms that exist, particularly in the gene that controls the beta chain of the hemoglobin molecule. We'll talk a little bit about what that is and how it relates to human evolution and advantageous reproduction. So remember that evolution occurs at the population level. A single breeding population is called a deme, and all of the genetic material in a deme or in a population is called the gene pool. So when we look at species, we're going to kind of take a look at how we define a species. And essentially the different forms of what we call speciation, or how new species are created. So how really do we define a species? Well, a species is defined on their ability to reproduce with one another, right? So if they can reproduce with one another, then they are in fact the same species. That's what's known as the biological species concept. So in terms of creating a species, you have reproductive isolation, and essentially this is one method in which you can create new species. Essentially you have a group that becomes isolated from the rest of the members of their species, and over time little differences within a group will accumulate, and essentially eventually those two two species will form out of one species, right? So eventually the group splits into two separate populations. So think of Darwin's finches in this case. So if we look at another example, we have emus and ostriches, which were actually ancestrally the same species, but as Australia, due to geologic plate tectonics, began to move away from Africa in the ancient past, they became reproductively isolated from one another, and slowly over generation after generation after generation of emus and ostriches, little differences accumulated between these kind of two groups, and then eventually they became so different as to be classified as two separate species. In terms of evolution, we have two types of change that we kind of measure. We have micro versus macro evolution. If we're looking at the changes in the expression of alleles from generation to generation, we are looking at micro evolution. If we are looking at large-scale changes such as speciation or the creation of a new species, that's changes in the skeletal structure in a single species, these type of changes take many, many generations to happen, and we refer to that as macro evolution. So you can think about it this way. If we're looking at the change in the carapace color for some beetles from one generation to the next, we're looking at microevolution. If we're looking at how horses became, started out as a creature that had fingers, essentially, or paws, kind of, to a creature that has a single hoof on each foot, we're looking at macroevolution. We also have cladogenesis, which is the act of a new species branching away from a common ancestor. So you can think about this as how there is a common ancestor for basically all dinosaurs, but then once, over time they began to diversify into many, many different groups. And this all relates back to extinction, right? Because it's essentially what we're looking at. All of the creatures that we are examining in the fossil record are no longer around, right? So we do have, anthropologists that study living humans, but in terms of biological and physical anthropology, most of what they do is looking at the fossil record, right? And in terms of evolution, that's what we want to look at as well. So extinction is defined as the loss of a species due to the disappearance of its members, and extinction is a common fate, right? Most species that have ever existed in Earth's history are now extinct, right? So there are more species that have gone extinct than there are currently existing. And some species, as we'll talk about later on in the semester when we get to kind of 10,000 BC, start to go to extinct due to gigantism, right? Their body sizes become too large, then there's a climatic shift, and their bodies are unable to kind of accommodate for that climatic shift. It happens too quickly for the evolution to really take, a hold and prevent the extinction of the species. So to give you a bit of a preview of what we're going to be looking at, we're going to look at how human variability is present everywhere in terms of skin tones, metabolic rates, biochemical traits like blood composition. Humans are, that's where we see a degree of variability, right? Differences in appearance are the result of environmental adaptations and outward appearance over time. We're going to look at the relationship between the sun and human skin. We're going to look at the relationship between heat and cold and how that relates to human adaptations, and thus why we have a large degree of physical differences on outward appearance and even metabolic differences. So remember last time we talked about the ABO blood group, right? Remember each of us has 2 alleles for this trait. So from one parent you might inherit an ABO and the other you will inherit either an ABO, right? So your genotype for this trait determines what your blood type is. Right, we talked about this in the last lecture. Just be sure to review this. In the ABO blood system, just remember that A and B are each dominant over O. So what you see expressed in the person, their phenotype, is due to the dominant recessive nature of those alleles. So if you have type A blood, your genotype is either 2 A's or an A and an O. Type B blood, it's either 2 B's or a B and an O. If you have type AB blood, you inherited both A and the B alleles, right? If you have a type O blood, you inherited the double recessive condition, having 2 O alleles from each parent. So if we look at another process, and the reason why knowing your blood type is important is because you don't want to get into a situation where you're being transfused with the wrong type of blood. Essentially what will happen if you receive the wrong blood type in a transfusion is that the antibodies within your body will start to react to the antigens that are on the surface of the blood cell, causing these blood cells to begin to become locked together in a process that's referred to as agglutination. And it turns your blood into kind of a jelly-like substance, which is not particularly good. It thickens the blood and can cause all sorts of deleterious issues and could cause death. Remember last time we talked about polygenic variation and pleiotropy. Much of this genetics today is based on a 1 gene, 1 protein model, but we know that many traits are polygenic and are determined by genes at more than one locus. The one that we're going to talk about is the beta globin gene mutation for sickle-cell anemia, and that's going to become very important in terms of a adaptation. Sickle-cell anemia and this mutation that we're going to see in the beta globin gene actually has an evolutionary advantage for the people who possess that mutation. Red blood cells carry proteins on their surfaces that function as antigens, right? Remember, those are what could react to the antibodies within your blood system. And antigens are any foreign substance recognized by the body, stimulates the production of antibodies. Antibodies are proteins produced as a defense mechanism to attack foreign substances invading the body. So agglutination is the clumping of red blood cells as a result of the reaction of antibodies to an antigen. So why do we have kind of polymorphisms and why do we have different blood groups? Well, it's actually these different blood groups have certain advantages, right? So why do we have these polymorphisms at that ABO locus? Well, the B allele actually helps protect against infant diarrhea. So if you're someone who was born with the B allele, you will actually have a slight protection against that disease, which in much of prehistory and even pre-industrial society, this is a huge killer of children. If you have both the A and B alleles that help protect against the plague, many people who had type AB blood actually survived the plague when it ravaged medieval Europe. If you have the O allele, it protects against bronchial pneumonia. If you have something called the RH plus allele, makes you more susceptible to smallpox. And this is part of the rhesus group, but we'll talk about that later. But it will make you more, and a lot of people, a lot of indigenous people of the New World, particularly Native Americans, have this Rh plus allele, which is why smallpox was particularly deadly to them. They were not exposed to the disease, and two, they didn't have any blood mechanisms that would help provide defense for the disease. So what we're looking at are disease pressures, right? Diseases exert significant evolutionary pressure on humans. It takes an awful lot of, deaths and things like that, for kind of natural selection to really kind of weed out people who do not, who have no resistance to these diseases, right? So you can imagine that over time, the people who would be most successful at reproduction and living longer lives will be the people who had the certain blood types that would offer them slight protection against these diseases. So when we're looking at evolution, and you may have to deal with this in your lab, we're not exactly sure yet, but you may see this pop up in your lab. It's the Hardy-Weinberg Equilibrium equation. It's an equation that will let you know if evolution is happening in a population. At most times, populations are in stasis, meaning that they're not changing a whole lot. If you know the frequency of certain alleles, you can predict the assortment of genotypes and phenotypes that would be expected if evolution was not occurring, if there was no change from one generation to the next, right? So the Hardy-Weinberg equilibrium model will indicate if genotype frequencies are deviating away from what is expected. So the purpose of the Hardy-Weinberg equilibrium equation is that we watch what happens to populations from one generation to the next. Do the frequencies of genes or the frequencies of alleles change, right? If they do, evolution is happening, at least, microevolution, right? So if we look at the steps of the equation, first you isolate your population, then you measure the allele frequencies for a particular trait. And then the genotype frequencies you observed are compared to the frequencies predicted by the equation. There are a few a priori assumptions. One, that your population is infinitely large. Two, that there are no mutations, meaning there are no new alleles from one generation to the next. There's no gene flow, no new alleles in, no old alleles out. No selection, so no alleles are getting weeded out from one generation to the next, and that there is random mating in the population. So in other words, nothing in, nothing out, and then random shuffling. So if all of these conditions are met, the allele frequencies will stay the same from one generation to the next, right? No evolution will occur, i.e. equilibrium, right? So we can compare these ideal or expected frequencies with the real frequencies that we discover when we examine the population, and this will let us know if evolution is happening. So all of this is an extension of the basic inheritance that was brought up by Mendelian genetics. So these baseline values provide a kind of way to measure species equilibrium, and it provides a way to measure the amount of change over time, right? And that's essentially what we're trying to do when we're looking at how evolutionary change occurs over time. So let's talk a little bit about mutations here. Mutations are the only source of new or novel genetic material in a population. If they're, essentially, for humans at least, most of us are 99.9% genetically similar to each other. It's really only a tiny, tiny bit of our genes that actually varies between one individual to the next. So it is really, mutation is the only real new source of genetic information. A mutation can be any heritable change in the structure or amount of DNA or genetic material. Changes can be as small as one base pair substitution, which is called a point mutation, or they can be as large as inheriting the wrong number of chromosomes, also known as a frame shift mutation. So if an allele's frequency, though, is 1% or higher, it's not just there because of a mutation, right? Some sort of evolutionary force has kind of latched on to that mutation and has started to perpetuate it within the population. So at this locus, we say there is a polymorphism, right? Some evolutionary force, such as natural selection, gene flow, or genetic drift, has increased its presence in the gene pool. So if we look at an example of just a simple point mutation, we have, and we're not going to get into the details of which gene controls all of this and how the mutation itself occurs, but just to give you an example of what the effect of a mutation could be, just as simple as a point mutation, there is a point mutation that affects the chloride channel on your cells, which could actually have some pretty deleterious effects if you are born with this mutation. It disturbs the movement of macromolecules into the cell. It can disrupt the ionic homeostasis as well as the acidification within the cell, which damages the organelles within the cell. In essence, what it does is it kind of disturbs the environment inside of the cells and makes it so that it's very hostile to all of the different little parts and pieces that operate within the somatic cells within your body. So the resulting pathologies that we see that come out of these kind of point mutations that affect the chloride channel are things like cystic fibrosis, chronic kidney stones, as well as Bartter syndrome. If we look at a frame mutation, on the other hand, this is a large-scale mutation, in essence, an extra chromosome, and it is not heritable if it occurs in males because it tends to make males sterile. So if it occurs in females, it's kind of a 50-50 whether or not it's going to make them sterile or not. Depends on which extra chromosome they inherit. So if we look at human polymorphisms that we see out in our own population, chromosome 11 carries the code for the beta chain of the hemoglobin molecule. The normal allele there is called hemoglobin A, or HbA. The sickle-cell allele is called hemoglobin S, which produces abnormal hemoglobin. And we'll talk here a little bit about kind of the effects of sickle cell and why sickle cell anemia and sickle cell disease exists in human populations. So in the video that you watched in the prior slide, you saw the initial life cycle of a parasitic disease called malaria. And the issue with malaria is that it's a pretty deadly disease for people who have no resistance to it, but there is something where you do have a resistance to malaria, right? This comes in this form of the HbS allele. The issue is that if you're born with 2 copies of this allele, you end up getting sickle cell disease and may die very early. But if you only inherit 1 copy of this allele, you in essence are slightly resistant to malaria, and we'll take a look at that, and I'll explain that further when we get to the actual pictures of the blood cells, so you can see it a little bit better. But the polymorphism here on chromosome 11 is the result of a simple base pair mutation. So if we look at the kind of the 2 normal alleles, 2 HbA alleles, you're homozygous normal. If you have 1 normal allele, 1 HbA, and 1 HbS, you are heterozygous, which means that you are malarial resistant. If you are have 2 copies of the affected allele, 2 HbS alleles, you are homozygous in the other direction, in essence, and you will be born with sickle cell anemia. So in essence, you want to kind of be a heterozygote in geographic regions in which there are high prevalence of malarial parasites transmitted by mosquitoes. So in essence, this HbA allele, it causes a substitution of an adenine with a uracil, right? In normal blood cell coding, you have a thymine to an adenine, which produces the glutamate, right? And the abnormal, or the HbS blood cell, adenine goes to uracil, which produces valine, which ends up affecting the structure of the red blood cell. So if we look at this in terms of kind of geographic regions in terms of malarial resistance, right? So if you think of Europeans, right, they are homozygous, they don't deal with malaria, it's not prevalent in their particular areas. So you don't have any type of evolutionary pressure or any selective pressure forcing that population to have a polymorphism at chromosome 11. But if you go into West Africa and some places, pretty much kind of the central band of Africa, there are high incidences of the malaria parasite, right? Because there's a lot of mosquitoes that carry the parasite in those geographic regions. So that's where we see this condition of the heterozygotes and the recessive homozygotes that have sickle cell anemia. This is just showing you how the substitution takes place in terms of how a normal hemoglobin versus a sickle cell hemoglobin is made. And the end result of which turns, it slightly misshapens the red blood cell, right? So what ends up happening in people that are heterozygous for this condition end up having a red blood cell that is only slightly misshapen, right? And it makes it impossible for the malaria parasite to really attach onto that red blood cell and to kind of reproduce and really kind of take over the system of the infected human. So if we look at HbS allele distribution and areas where malaria is present, right, you can kind of see that it's a direct correlation, right? We can overlay those two maps together and see that in areas where malaria is high, we see large percentages of this HbS allele distribution. But there is a trade-off to this, right? Because having a normal red blood cells means your red blood cells are slightly aerodynamic, right? They can move through your arterial system without any problems. But having a sickle-shaped cell or having a cell that's slightly misshapen could potentially lead to vascular problems later on down the road, right? And the reason that is because the red blood cells aren't as aerodynamic. They may get kind of caught up in different parts of the arterial system, leading to blood clotting, which could lead to heart attacks and stroke. And the interesting part about this, and what you have to remember in terms of evolutionary theory, is that the reason we can get rid of all of the mosquitoes in these areas, we can stamp down malaria, which, in a lot of degrees we've had some success doing that in places in Africa. But the reason why this HbS allele doesn't disappear out of the population is simply because it does not affect the individual before they are capable of breeding and passing that gene on to the next generation, right? These heart problems and these potential vascular problems that we're talking about that comes with being a heterozygous for the HbS allele actually really doesn't occur until later on in life. But if you receive both affected alleles, you end up being born with full-blown sickle cell anemia, which comes with a whole host of different kind of symptoms and conditions that can develop, everything from painful osteomyelitis to chronic ulcers. You could have incidences of stroke or cardiovascular events. You could have avascular necrosis. There's, like I said, there's just a whole host of different problems that an individual born with full-blown sickle cell anemia has. There are other polymorphisms at chromosome 11. We have G6PD deficiency, or otherwise known as favism. It's an X-linked recessive disorder characterized by red blood cell hemolysis after exposure to oxidative drugs, things like aspirin, sulfonamides, dapsone, as a few others. Fava beans will actually, this is why they call it favism, or an infection could cause this. In most cases are self-limited as red blood cells are replaced, and oxidative drugs should be avoided in these individuals. But these individuals have red blood cells that are slightly misshapen that makes them resistant to malaria, right? So we see G6PD deficiency in malarial areas as well. So what happens in this case? It's an X-linked trait, right? So if you have G6PD deficiency, it can cause an oxidation of sulfohydryl proteins within the cell. The oxidation causes the proteins to become denatured, and the result is insoluble cellular detritus called Hindbody, right? So the resulting pathology is called hemolytic anemia. So in essence, what happens if you have G6PD deficiency and you take oxidative drugs, in essence, you could be destroying your somatic cells by essentially creating this hostile environment for the cell, and you get this nasty garbage that builds up and just kind of sits inside the cell and causes damage. So this is showing you how the evolutionary, or kind of how it would be passed from one generation to the next. So just make sure you kind of review this chart and look at the conditions. If you had one who was affected and one unaffected, you're going to end up having two carrier children. If you have an unaffected and a carrier, you're going to end up having one carrier and one affected. There are other polymorphisms. We're not going to get into much detail about thalassemia, but it's just another polymorphism on chromosome 11 that slightly misshapens the red blood cell and makes those individuals resistant to blood-borne parasites like malaria. The severity of thalassemia varies. You have severe thalassemia, also known as alpha thalassemia. It's an abnormal metabolic function, which essentially results in lower than normal blood cells. Or you can have mild, or what they call beta thalassemia, which are just abnormal levels of blood cells. They're just only slightly abnormal. And the severity really depends on how many factors are inherited from the parents or lack thereof. So symptoms of thalassemia or alpha thalassemia, you can actually have jaundice in babies. You can have an extremely enlarged spleen due to kind of some overworking, and your cheeks, forehead, and jaw may all overgrow. But what we're looking at with all of these conditions is something that we call balanced polymorphism, right? Where the heterozygote individual has the greatest fitness and the greatest reproductive success in these environments, right? But they must be careful in reproducing because, they will inevitably, if they're mating with another heterozygote, will produce a child who is affected by full-blown sickle cell anemia. So natural selection, based on Darwin's principles that individuals with advantageous characteristics will survive and reproduce in higher numbers or higher reproductive success, right? That is the process of natural selection. So Darwin, in essence, never really said the phrase survival of the fittest. That's kind of been coined or assigned to him, but that phrase was actually not coined until 50 years after Darwin's publication by a completely different individual, and actually another anthropologist. What Darwin did say is that it is not the strongest nor the smartest creatures that survive, but those most responsive to change. There are different patterns of natural selection that we'll talk about here. There are three different types. You have directional natural selection, which favors an extreme form of a trait. You have stabilizing selection, which favors the average form of a trait. And then you have disruptive selection, which favors individuals at both extremes. So if we look at directional selection, if we look at an example, the increasing size of mammoths and other mammals during the ice ages can be seen as directional selection. The environment was very cold and pushing them to become larger bodied. The increasing size of the human brain over the last 2 million years is a form of directional selection. Stabilizing selection, that's what favors the average form of a trait, and living fossils are a great example of stabilizing selection. Here's a horseshoe crab that's about 5 million years old. There's a horseshoe crab that's about 3 years old, right? You can see that there's not a whole heck of a lot of change that's occurred in that species over time. Disruptive selection favors both extremes of a trait, and if it goes on long enough, it will actually create new species, right? So think about Darwin's finches. So another example of disruptive selection can occur with the African swallowtail. The male or female is palatable and does not mimic any unpalatable species, right? The female, but you do have females that are born that will mimic an unpalatable species, and you will have females that are born that mimics a different unpalatable species, right? So it really, you start to see a favoring of those two individuals on each side, and the individual in the middle, which is kind of the ancestral version or the original version, starts to kind of cease to exist or starts to decrease in numbers over time. If we look at another example of natural selection in action, we have the case of the peppered moth, right? We actually see two peppered moths here on this tree. One of them is kind of closer to what the original peppered moth looks like, that speckled one down at the bottom. And the peppered moth you see up in the top that seems to blend in more, that one is kind of the peppered moth that we see more commonly today, right? So, and this really relates to kind of an interesting thing that happened during the Industrial Revolution in England, right? As the Industrial Revolution kind of kicked off and these industries were burning more coal, you had a lot of coal soot that was becoming deposited in England, and particularly it would become deposited on the barks of various trees, right? So the peppered moths who would cling to the side of these trees that were ended up being born with darker coloration blended in with the trees more and were able to survive. The original moths which were kind of more of a light color, which were meant to blend in with the kind of lighter colored birch bark, were not surviving because they stuck out like a sore thumb against that darkened tree bark. So this is just showing you that, and it kind of shows you, Manchester, Birmingham, East Anglia, these are all very heavy industrial areas. If you look at Dorset, that's actually a more rural area. So you can actually really see how, it sits at it. This kind of shows you an interesting intersection between human interaction with the environment, how humans can affect the environment, and how that in turn affects local species that live within the environment. So what type, ask yourself what type of natural selection is seen in the case of the peppered moth? If you guess directional selection, you are correct. To talk about sexual selection here for a little bit, it's another form of natural selection. It's natural selection arising through the preference by one sex for certain characteristics in an individual of the other sex. And the interesting part about this is that sexual selection can sometimes go horribly wrong, right? The Irish elk actually went extinct due to sexual selection because females began preferring males that had larger and larger horns, and the competition between males became fiercer and fiercer, and that also drove subsequent generations to be born with larger and larger horns, which ended up becoming a little cumbersome, and in a lot of cases these individuals became locked together when they would kind of ram each other in competition, and they would both end up dying. Right, so what type of selection is sexual selection? And why do we have sexual selection, right? Well, inevitably, why do we have sexual dimorphism, right? These are all things, you should consider when you're looking at the evolutionary history of any given species. So what types of competition are there, right? Well, of course, we have violent competition, but we also have other forms of competition that we'll kind of talk about here. In primates, which, as we move through the semester, we're going to focus a lot on primates and then on kind of our own species. But there is a selective force in gorillas towards small testicles. And this is because in their evolutionary past, gorillas used to kind of more violently compete with each other over access to mates. And what would end up happening is, one of the first things the male gorilla would go for would be the other male gorilla's testicles, right? So in essence, over time, those gorillas that were born with slightly smaller testicles were actually able to be more successful in breeding because they were able to kind of get through this competition process without losing their reproductive organs. So studies show larger testicles in gorillas also lead to unfaithful male gorillas, right? And of course, we just discussed the violent competition that occurs between males as a causal factor as well. But in other primate species like chimpanzees, we have sperm competition, right? Which is actually really an interesting process, right? Since male chimpanzees, since they live in small groups, there's not a whole lot of violent competition between males. So they will compete in kind of another way. They'll compete in kind of sperm production, right? Whoever kind of produces the most sperm tends to be more successful in copulation with the group's females, right? So the question is, since we are so highly related to chimpanzees genetically, does sperm competition occur in humans? It actually does, right? And this is based off of kind of the variation that we see in human sperm type, right? Some humans are born with more efficient, for lack of a better way to put it, some humans are born with more efficient swimmers. Some humans are born with less efficient swimmers, as well as different variants and levels of production. So if we look at another form of our evolutionary force, genetic drift always acts to decrease variation within a group. It's more common in endogamous groups within humans, or humans that marry within their own kind of social and kind of genetic group. So here we can see just kind of gene frequencies changing over time, and you can see B represents genetic drift, and you can see the frequency starts to decrease dramatically over time. C represents kind of a stabilizing selection, and A representing a more natural selection. And this could lead to something called Founder's effect. An example in our text that they use is hypercholesteremia that exists in South Africa. So in essence, what you had happen is in area A representing Europe, a small group of Europeans that had the kind of alleles that led to increases in hypercholesteremia left Europe, went to South Africa, and began spreading those alleles through the South African population after, in successive generations. Genetic drift is common in insect species, right? Newly founded populations do not always represent the genetic diversity of their sources. Another evolutionary force, gene flow, always works to increase genetic variation within a population. And a very easy way to kind of think of this is, let's say you have two groups of humans, one with blonde hair and one with black hair, and they begin to marry each other or breed with each other, and in the next generation you have children that are born with both black hair and blonde hair, right? That is what gene flow is, the exchange of those genes. So in essence, it's kind of like this, right? If we go back to, Darwin's finches, right? We also see in our evolutionary history, we can see the human gene flow of type B blood. The B blood type distribution, or the kind of, if you look at it on the chart here, the allele no doubt originated as a mutation and then spread westward through gene flow. So we can actually map different kind of genes that have kind of moved through different populations over time. So let's look at mutagens. We already talked about mutations a little bit, but this is kind of going to be my PSA section about kind of the way in which we run our society and how many mutagens you actually live around on a daily basis that are altering your DNA. And there are different categories of mutagens. There are physical mutagens, there are DNA reactive chemicals, there are base analog mutagens, intercalating agents, which are mutagens, and heavy metals, which are mutagens, as well as viruses. So if we look at some physical mutagens, X-rays are a physical mutagen. You need a heck of a lot of X-ray exposure to cause a mutation. UV radiation is also a physical mutagen, but you also need a lot of exposure. This is what can cause skin cancers. Radioactive decay of carbon-14 is also a physical mutagen. And certain industrial byproducts are physical mutagens. This is actually shows you a burial pit where they are burying radioactive materials, right? And the problem is that eventually all that stuff is going to decay, and when it does decay, it is going to eventually leach into the surrounding soils, which could potentially pose an environmental problem. If we look at DNA reactive chemicals, we have PAHs, which is polyurethane foam, which is found in a lot of cushions and kind of bedding and things like that. It's a fire retardant. We have automatic or aromatic amines, which are found in things like plastics, household cleaners, as well as grilled meat. These can all cause mutations in your DNA. These chemicals do not cause mutations in everyone, and the mutations are the byproducts of the body's metabolic activity, are not uniform across all people. That's why we still allow PHAs and aromatic amines in these different kind of products and things that we use in our daily lives, right? Because it does not cause mutations in everyone, only a small group of the population. So look at base analog mutations. These are the point and frame shift mutations that occur naturally over time. So far, these are the only type of mutations that have actually contributed to the human gene pool and still exist today. So remember our HbS allele we talked about earlier. This is a good example of a base analog mutation. Intercalating agents, things like proflavine, which is a hospital cleaning chemical, acidium bromide, which is used in molecular biology labs. Direct contact with intercalating agents can cause fatal frame shift mutations over time. If we look at metals such as arsenic, cadmium, and nickel can cause mutations if you have long-term exposure. Nickel will disrupt your DNA repair mechanisms. Cadmium will cause DNA mismatches during somatic cell replication. And arsenic, large exposure, of course, causes death. Small exposure causes an increase in free radicals within your body, which damages cell function, right? So it's, in essence, too much exposure to these could cause a whole host of mutations, which could lead to different cancers. If we look at viruses, the flu, HIV, as well as smallpox, all three of these have actually caused or can cause changes in your DNA if they are left kind of unchecked within your body. So let's just provide just a few examples here just to kind of cement in the concepts of population genetics and evolution at a population level. If we look at natural selection, to give an example, a species of rats live in a certain type of tree where the branches are evenly spaced. Smaller rats could not reach from branch to branch, and larger rats would break the branches and fall and die. So soon, all of the rats, after a few generations, were just the right size for the tree branches, right? That is natural selection in action, choosing the most fit for the environment. If we look at gene flow, we have a bunch of women from West Africa where malaria is present. They mate with a group of Europeans. Their children are less susceptible to contracting malaria due to the presence of antibodies from their West African mothers, right? So in essence, this is a form of gene flow. If we look at genetic drift, a man steps on a group of beetles, randomly killing most of the green ones, but leaving most of the brown ones alive, resulting in fewer green beetles being produced in the population in the next generation. And then finally, remember your two different types of mutations, right? You have a point and frame shift mutation. A point mutation is a switching of a single nitrogen base within DNA, generally the least harmful of mutations, and some mutations have no effect on the protein being constructed at all. You then have frame shift mutations where an entire nitrogen base of DNA is cut out or duplicated incorrectly. You have examples of this like Crohn's disease as well as IBS, or irritable bowel syndrome.